Welcome to the deep dive. Today, we're really getting into the weeds, you could say, or maybe the kernels. We're going microscopic, talking about plant stem cells. These tiny things are, well, fundamental. They produce almost everything we rely on, our food, feed for livestock, even renewable fuels. So our mission today is to unpack some really groundbreaking research on these cells. We know stem cells are the foundation of plant growth, right? Every leaf, every root, but how they're controlled, that's been a bit of a mystery.
Exactly. It's kind of wild when you think about it.
Yeah.
These are the growth engines for, well, basically the planet's food supply. But yeah, despite knowing how crucial they are, getting the full picture has been tough. Past studies, they really struggled. They could only pinpoint a few key genes, the ones running the show. It's like trying to understand a whole factory floor when you only have like half the circuit diagram.
Yeah.
It just doesn't work.
Right, you've got the structure, the factory itself, but the actual control panel blueprints are missing huge chunks. And you can't really optimize production or, build better factories without those blueprints. So who decided to finally tackle this head on?
That leads us to Cold Spring Harbor Laboratory. Professor David Jackson and his team, they took this on. Their goal, I think this sums it up perfectly, was to get the knowledge needed to, quote, know how to make a stem cell.
Wait, know how to make a stem cell. That sounds, well, profound, like finding a fundamental secret of life. What's the big payoff? I mean, why is this specific knowledge so critical right now?
Well, think bigger picture. Climate change, food security. The applications are massive. We urgently need crops that are, as Jackson put it, more resilient or more productive. If you understand how a stem cell works, how it starts, stops, regenerates, you can potentially engineer plants to better withstand drought or heat or disease. It's about stabilizing our food systems. Jackson realized that to get past just, trial and error breeding, they needed the whole list, the full list of regulators, the genes controlling growth. That was the mission.
The fullest. Okay. And that brings us right to the core problem, doesn't it? These cells are fundamental, yes, but they're also incredibly rare and hidden deep inside the growing tips of the plant, the meristem. You can't just harvest them.
Oh, absolutely. The access issue is huge. You're talking maybe a few hundred active cells in an entire plant sheet. Very, very few. So they needed a breakthrough, not just in analyzing the genes, but in actually getting to the cells first. Yeah. They started with two regulators that were pretty well known, clavata-3 and WUSCHEL.
Okay, let's just quickly touch on those. For listeners who might not be familiar, what do clavata-3 and WUSCHEL actually do?
Good point. Think of them like the gas pedal and the brake for stem cell production. Clavata-3 is basically the brake. It sends a signal saying, okay, stop making more stem cells here. Let's make a leaf or a flower instead. And WUSCHEL, that's the accelerator. It tells the cells, stay as stem cells, keep dividing, keep the potential going. They're in this constant push and pull, this delicate balance.
Yes, and brake. Got it. So the goal was to find all the other pedals and levers in that control system. But first, they had to physically isolate those rare cells that were using the known gas and brake pedals.
Right. And that involved some really meticulous work. A former postdoc, Shasa Shu, had to very carefully dissect these tiny, tiny sections from the growing shoots of maize and Arabidopsis.
Arabidopsis, that's that little model plant scientists use a lot, right?
Exactly. A workhorse for plant science. So after this incredibly careful dissection, they used some pretty advanced tech to handle these rare cells. This is where the real innovation technique comes in. They used a microfluidics device.
Microfluidics? Like tiny pipes?
Sort of, yeah. Imagine a microscopic plumbing system on a chip. It forces the cells through tiny channels, separating them one by one.
Wow, okay.
And once they were separated, they applied single-cell RNA sequencing, or SCRS.
SCRS. Okay, this sounds like the crucial bit for getting the data. How does that work? Giving each cell its own voice, you said.
That's a great way to put it. So RNA is like the cell's current instruction manual, what it's actively doing. They took the RNA from each single isolated cell and converted it back into DNA. And here's the clever part. They attached a unique molecular tag, like a barcode, to that DNA.
A barcode, so you know exactly which cell that specific piece of DNA, that instruction came from.
Precisely. It's like putting a tiny, unique GPS tracker on the activity of every single cell.
That's incredible, because usually you're just looking at the average activity of thousands or millions of cells all mashed together, right?
Exactly. This gives you unprecedented resolution. You see what individual stem cells are doing.
So they could map gene expression across thousands of single cells simultaneously?
Yes. And the amount of data they got was pretty remarkable. They managed to recover about 5,000 cells expressing clavata-3, the brake, and about 1,000 expressing WUSCHEL, the accelerator.
5,000 and 1,000, considering how rare these cells are, that sounds like a massive success in just isolating them.
It really was. It turned what seemed almost impossible into a solid data set to work with.
And that data, that was the key, unlocking the next step. So they charted these known regulators. What else did this super high definition map show them?
Well, it wasn't just about confirming the brake and the accelerator. That was important, but the really exciting part, they found previously unknown regulators. Lots of them.
Ah, so new pedals and levers for that control panel.
Exactly. They identified hundreds of new genes that were switched on or expressed much more, specifically in the stem cells of both maize and Arabidopsis.
Both. But maize and Arabidopsis are really different plants, aren't they? One's a grass, one's more like a weed.
Very different. Maize is a monocot, Arabidopsis is a dicot. They diverged evolutionarily a long, long time ago. So finding hundreds of the same genes active in the stem cells of both, that's huge.
Why is that so significant? What does it suggest?
It suggests these regulators aren't just random bits of code specific to one plant type. They're likely evolutionarily conserved, meaning they're probably fundamental and important across a vast range of plant species. It's like finding a common ancient language that many different plants use to control their basic growth.
A universal plant language for stem cells. Wow.
Yeah, and this fundamentally changes things. Researchers looking at, say, rice or wheat stem cells might not have to start from scratch mapping everything. They can now look at this list of conserved genes and say, okay, let's test these first.
That speeds everything up dramatically, like having a cheat sheet.
Exactly. And the team made sure everyone could use it. The result of all this mapping is what they called an atlas of gene expression.
An atlas.
Yeah, a detailed map. And Jackson was clear. This atlas is being published, made available for the whole community.
So other scientists, breeders, biologists, they don't need to repeat this incredibly complex, difficult experiment themselves.
Nope. If you're working on maize or Arabidopsis stem cells, the foundational map is already there. You just open the atlas.
That's a massive contribution. It really could accelerate research globally.
It really could. Potentially shave years off discovery timelines. Yeah. Now let's bring this back down to earth a bit. Back to the farm, the field, the future of the harvest, because this wasn't just theoretical.
Right, there was a direct link to practical agriculture.
Yes. Almost immediately, they found connections. They were able to link some of these newly identified stem cell regulators directly to differences in size in maize plants.
The size differences, you mean like bigger corn cobs?
Yeah.
Higher yield?
Essentially, yes. Traits that directly influence productivity. They could see a genetic connection between certain regulators and, for example, the dimensions of the corn ear.
Okay, so if you can tweak the regulators, maybe you can influence the yield.
That's the potential, yes. But it also raises that classic question, right? Is bigger always better?
Ah, the trade-offs.
Exactly. If you push for maximum size using this map, what else changes? Nutrient density, water needs, disease resistance.
So the breeders have this powerful new map, but they need to navigate it carefully. Balancing yield with resilience, maybe?
Precisely. But the key thing, the immediate so what, is the precision it offers. Breeders can now select specific strains with much more targeted goals.
How so?
Well, maybe you need a maize strain that's super drought tolerant. It might yield a bit less, but it saves water. Now, you might know which specific regulators to look for or target for that trait.
Or if you're growing maize purely for biofuel, where it's all about biomass, just sheer size.
Right. You could select for regulators that maximize that, maybe sacrificing something else that doesn't matter for fuel production. It allows for tailored engineering, not just broad selection.
Targeted engineering. That sounds like the future. The lead researcher, Jackson, called this foundational knowledge guiding research for the next decade. It's not just a quick fix.
No, definitely not. It's building the bedrock understanding. He pointed out it benefits like three main groups. First, developmental biologists, one studying the basic how does a plant become a plant questions.
Okay.
Second, physiologists. They study the whole system, how corn ears actually develop, how nutrients move, how to boost overall productivity. This gives them genetic levers to investigate. And third, of course, the breeders, the ones actually designing the crops for tomorrow's climate. This atlas is a core tool for them. It enables real precision agriculture at the genetic level.
And that precision, that understanding is what we desperately need for global food security, isn't it?
Absolutely. Mapping these tiny pathways is directly linked to stability. It helps ensure future harvests can be more predictable, more robust, especially with, all the environmental changes happening.
It moves us away from just hoping traditional breeding keeps up towards actively understanding and potentially guiding plant growth.
It gives us the language, you could say, the language of the plant's own growth engine, so maybe we can communicate with it more effectively.
So let's leave our listeners with this thought. Jackson's team developed this amazing technique to isolate really rare cells in maize and Arabidopsis. What if, as they suggest, this technique could be applied broadly across many plant species? Well, think about staple crops: rice, wheat, sorghum, plants feeding billions. If we could create similar atlases for them, understand their stem cell regulators, what kind of resilience could we unlock? Maybe against rising sea levels impacting rice paddies or chronic drought hitting wheat fields? The potential seems, well, enormous. Truly enormous. Thanks for joining us today for this deep dive into the building blocks of our food supply. We'll see you next time.